Plant hormones are needed in small amounts, and there are 5 groups of plant hormones or plant growth regulators. You'll see that I use the term interchangeably. So auxin, cytokinins, gibberellic acid, ethylene and abscisic acid are proven hormones or plant growth regulators. These have effects at micromolar concentrations. There are also a range of compounds that have plant growth regulator-like activity, but are not universally acclaimed as plant hormones because they tend to be needed at much higher concentrations. If you look at auxins, there is only one compound, indole acetic acid, which is absolutely necessary in all plants. So these are absolutely essential. There are no mutants known. In the 1970s, a range of temperature sensitive mutants of auxins were produced, but these were very difficult to maintain and have since been lost. So at the moment there are no mutants for auxins. So it's possible to knock out a lot of gene functions by putting in antisense RNA. But you can't do this for auxins because they have essential functions in the plant. Now, although there is only one compound, indole 3-acetic acid, which is found in nature, there are many synthetic analogues like naphthalene acetic acid, NAA, indole butyric acid, IBA, and some analogues that have a benzene ring in them, 2,4-dichlorophenoxyacetic acid, 2,4-D, 2,4,5-trichlorophenoxyacetic acid, 2,4,5-T, and herbicide pichlorum. And these tend to be cheaper and more stable, but they do have some disadvantages, particularly 2,4-D, 2,4,5-T and pichlorum. They're generally growth stimulatory and generally promote rooting. They're produced in meristems, especially the shoot meristem, and transported throughout the plant in special cells, in cells which are adjacent to the vascular cambium in the vascular bundles. Now, I would like to say right at the start that there are 2 distinct groups of synthetic analogues. Naphthalene acetic acid and indole butyric acid are transported in exactly the same way as indole 3-acetic acid in the auxin transport system. 2,4-D, 2,4,5-T and pichlorum are not recognized by the auxin transport system and are not transported throughout the plant in a polar manner. So for example, for micropropagation, where you want to maintain polarity in the plant, you would typically use indole acetic acid, IAA, or NAA or IBA, and you would almost never use 2,4-D, 2,4,5-T or pichlorum. If you want to induce undifferentiated growth in your plants to produce what's called a callus, which is an unorganized mass of cell, sometimes you'll use indole 3-acetic acid or NAA or IBA, but sometimes the polarity in the plant is so strong that the auxins are actually transported away from the site of action by the auxin transport system. And in that case, you may get better results by using 2,4-D, 2,4,5-D or pichlorum. But 2 discrete groups of synthetic auxins. Cytokinins, again, are absolutely essential. There are no mutants known. And there is a single natural compound, zeatin, which is transported throughout the plant in the phloem as the zeatin riboside. So if you're doing plant cell culture, you can use zeatin riboside, or you can use a number of synthetic analogues such as benzyladenine, BA, or kinetin. They are generally growth stimulatory, and they tend to stimulate cell division in conjunction with auxins. And cytokinins are essential for the formation of adventitious shoots. In micropropagation, most of the time, 90% of the time, we are trying to get outgrowth of axillary buds. Axillary buds are quite distinct from adventitious shoots. Axillary buds are found in only one place, the axil of the leaf. So wherever you've got a leaf coming off the plant, you've got an axillary bud. And outgrowth of this can be stimulated by explanting the plant, removing the axillary bud from the influence of the shoot tip. It can also be promoted by cytokinins, which will tend to counteract the inhibitory effect of the shoot tip on the outgrowth of axillary buds. But if you've got an explant that's got no buds in it at all, you can still get shoots formed de novo, and these shoots that are formed de novo, so you get shoots formed where there was no shoot initial there before. So basically you fool the plant into cell division and those cells into actually forming a bud, and these buds will then grow out to give what are called adventitious shoots. They're formed adventitiously where there was no shoot initial there before, and this was promoted by cytokinins. Gibberellins are a family of over 70 related compounds. Commercially, two forms are available, GA3 and a mixture of GA4 and GA9. They stimulate etiolation of cells, of stems, that is, they make the stems go longer, particularly in dwarf variants, and they help break bud and seed dormancy. Sometimes you'll see these being used for micropropagation to enhance the growth of the outgrowth of the axillary buds and promote their etiolation. They also tend to stimulate cell division. And again, they're produced in young leaves, so when you make an explant you're chopping away the young leaves, so you may need to supply these GAs. Ethylene is involved in the wound response in plants. It's produced in all cells of the plants, and it tends to produce thickening in stems and leaf abscission. This is not something that you want in plant cell cultures. It also reduces adventitious shoot formation. So basically in plant cell culture we tend not to add ethylene, but we tend to try and counteract the effects of ethylene. Now this is possible because we know that ethylene interacts with the ethylene binding protein in the cell membrane of cells, and this can be inhibited by compounds like silver nitrate or organic compounds like norbornadine. Now these bind to the ethylene binding protein and they antagonize ethylene effects. As well as compounds which are broadly stimulatory to growth, like auxin, cytokinin and gibberellin, there are also some compounds which are generally inhibitory to growth. One of these is abscisic acid. Now there's only one natural compound. It was discovered in the 1960s by two groups, one working on leaf abscission, that is the way that leaves fall off in winter or in autumn, and seed dormancy. And they independently isolated a compound. Those working on leaf abscission called it abscicin, those working on seed dormancy called it dormin, and they eventually realized it was the same compound. So by mutual agreement they agreed to relabel it abscisic acid. It also plays a dominant role in closing stomata in response to water stress, so it's very important physiologically. From a plant tissue culture point of view, it has an important role in embryogenesis, in preparing embryos for desiccation. It also helps to ensure normal embryos. So in embryogenesis, in somatic embryogenesis, we typically would try to propagate the embryos themselves. So you end up with secondary embryos growing on the surface of embryos. And for embryo development we want to stop this, and abscisic acid has an important role there. There are also plant growth regulator-like substances. Now these are substances that have plant growth regulator-like activity, but they're effective at much higher concentrations than plant hormones. So plant hormones would typically be effective at micromolar concentration, whereas plant growth regulator substances may be necessary at millimolar concentrations, so 1,000-fold increase. So there are compounds like polyamines, which have a vital role in embryo development. I'll be talking much more about them when I talk about somatic embryogenesis. Jasmonic acid has been shown to have an important role in plant wound responses, and salicylic acid, isolated from willow, also has an important role in plants. But these are not universally acclaimed as plant hormones, since they're usually needed at high concentrations. There are also a range of undefined supplements, which are sources of hormones, vitamins and polyamines. So for example, coconut water is very widely used, which contains, for example, high concentrations of polyamines, also some plant hormones like cytokinins, a range of vitamins and nucleic acids. And it can be very useful in helping plant tissue cultures. The big problems with these is that they're not reproducible. So what I normally do is, if I'm going to use coconut water, I would buy in a whole load of coconuts, produce a large amount of extract, and then I'll freeze it. And it'll keep for years. But they're not reproducible, although they do work.